Technical animation of cultivated meat. Cultivated meat is genuine animal meat that is produced by cultivating animal cells directly. This production method eliminates the need to raise and farm animals for food. Tissue sampling. The process starts with tissue sampling from a donor animal. After cleaning the sampling area, a local anesthetic is applied to the animal. Muscle tissue fragments are extracted, then stored and transported to the processing site. Cell isolation. With the muscle sample in hand, it's time to isolate the cells. To do this, the muscle fragments are dissociated, digested, and centrifuged to obtain a cell suspension. This suspension is transferred to culture flasks, where the cells adhere to the surface of the flask and proliferate while consuming nutrients from the media. Primary cell culture and immortalization. Once the cells cover the surface of the culture flasks, they are detached, split, and transferred into new flasks in a procedure called subculturing or passaging. Cycles of subculturing are repeated until a sufficient number of cells is reached. To enable large-scale cell culture and reduce the need for repeated tissue sampling procedures, immortalization can be performed. In approaches for cell immortalization, the mechanisms that lead to the slowing down and arrest of cell proliferation are overridden, enabling the cells to multiply indefinitely. This leads to the generation of immortalized cell lines, in which the cells maintain their proliferative capacity. Cell banks. As soon as a relevant cell density is achieved, the cells are frozen and stored in cell banks, where they are distributed in individual tubes and stored in ultrafreezers at temperatures below -150 degrees Celsius. The sizes of cell banks can vary depending on the type of production, but they are generally made up of hundreds of tubes containing millions of cells each. To ensure that the cells are suitable and safe for cultivated meat production, quality tests are carried out regularly to detect contamination and guarantee genetic stability, as well as other tests. Bioprocess. The production process begins by retrieving a tube of cells from the cell bank. These cells are thawed and cultivated until a sufficient number of cells, called the inoculum, is obtained to seed the main bioreactor. This progressive growth, obtained by an increasing scale of the cultivation system, is known as a seed train. The number of subculture steps in a seed train depends on factors such as the cell growth rate and the yield. For bioreactor cultivations, small porous structures known as microcarriers may be used to allow cells to attach and grow. Throughout the process, parameters including temperature, pH, and dissolved oxygen and carbon dioxide concentrations in the media are assessed to automate controls and monitor the culture in real time. Differentiation. Once proliferation is complete, the cells may be induced into a differentiation phase, which results in the establishment of cell types such as muscle cells, fat cells, and other support cells that make up the final product. This process can be triggered, for example, by changing the culture media. Components added to the differentiation media interact with the cells, guiding them from an undifferentiated state to a differentiated one, in which the cells show specialization. Muscle cells are specialized in contracting, for example, so differentiation into this type of cell includes the acquisition of a phenotype, internal structures and organelles that allow muscle contraction. Harvest. After differentiation, the culture media is drained, and the cells are washed and recovered as biomass by centrifugation or filtration. They are then taken through a series of steps to formulate a final product. From this stage on, the entire process is carried out at refrigeration temperatures. As with the processing of traditional meat products, this is important for guaranteeing the safety and quality of the product. Processing, hamburger root. After harvesting, the cells can be used in different ways depending on the type of final product you want to obtain. If the final product is unstructured, like a hamburger or sausage, the cells are used as one of the product's ingredients, similar to conventional product processing. Once harvested, the cells are mixed with other ingredients to obtain a homogeneous mass. The mass is shaped into the right size and thickness, and the finished patties are frozen and packaged. Whole-cut meat root. If the final product you want to obtain is structured, such as a whole cut of meat, chicken breast, or steak, additional structuring steps are required. The structuring of meat cuts can be done using different strategies, which generally involve the use of some type of scaffold, for example, porous, fibrous, hydrogel, or bioprinted. Want to know more about cultivated meat? Access free resources on our website, gfi.org.